Okay, hello everybody. I was told that I have to start as soon as the spotlights hit me, so that happened, so we can start now. Okay, I think that when you look at the title of this session, the value of data in medicine, so I think that in every meeting, in every session, also in this conference, you come to this conclusion anyway, and I think that could be really the title of the whole conference as well. But of course, at one point you have to be, have to start to become more specific. And I hope that during this session we can really present the several cases where we show that different data and then also data from different sources are really big value in the processes we are trying to achieve to really make all the health process more efficient and better. So today we have 3 speakers, and unfortunately Olli Kalaniemi couldn't come here, and that also left me alone here really leading this session, but we have another speaker from Sweden, and I'm really happy to start with Oliver Billker, who to my knowledge is a participant of the National Program for Data-Driven Life Science Steering Group, also the director of the Laboratory for the Molecular Infection Medicine Sweden. And if I'm wrong, please correct me, but the floor is yours, and we are waiting for your presentation.
Thank you very much. I hope you can hear me. And now I can still hear the echo from the room, but not anymore. Perfect. So Olli Kalaniemi sends his apologies. He regrets deeply that he cannot be with you, not even virtually, because he did not only catch a cold, but he also lost his voice. So he asked me late yesterday to step in and present his slides, which I'm now going to do. And I give you just a little bit of background about myself. I'm a professor of biotechnology at Umeå University, and I'm now going to switch my own sound off to avoid the echo. Okay, and I am also director of molecular infection medicine Sweden, indeed. So MIMS is the Swedish EMBL node. Just to give you a little bit of background, we are part of a Nordic EMBL-Partnership with our partner institutes in Helsinki, Oslo, and Aarhus. Just signed a renewal for the next 10 years last year. And we are, of course, very much aware of the fact that also the Baltic countries are increasingly engaged with EMBL, and part of EMBL and nodes are emerging in the Baltic countries, and we much hope to intensify our collaborations from a Nordic to a Baltic and a dimension in the near future. But that's just an aside. My support for DDLS is something I do in my spare time as a member of the steering group. And I moved to Sweden, just to give you that bit of more background, 5 years ago, having been a group leader at the Sanger Institute in Cambridge previously, and spent most of my life as a malaria researcher in the UK. So I come from a data-rich, technology-driven environment, and I was absolutely blown away by the Swedish answer to some of the challenges of the technology revolution in research, which is, of course, that no single institute, not even the Sanger Institute, and certainly no university, can hope to always remain at the cutting edge of technology and of all different aspects of technology. And that is, of course, where SciLife Lab comes in as an answer. We are now moving to a data-driven revolution, and even more recently, analysis using AI comes in. But in many places, all these changes have happened within an unchanged academic ecosystem, and with the same translational and product development processes, and in the same regulatory environment. And of course, yeah, SciLife Lab was the Swedish answer to changing the environment to the needs of changing technologies. And that is something I want to give you just a little bit of background on to begin with. This is just illustrating that in 2022 alone, 1,800 scientists worked on 3,700 projects across 10 technology platforms set up by Scilab Lab in 40 different units that are spread across the entire country, and which together have created in that year 10 petabytes of data. This infrastructure is largely used by national academic researchers, but not only. As a national infrastructure, this Scilab Lab can now manage to remain at the cutting edge of a large number of technologies. This just lists a few of the Nobel Prize winning technologies or Nature Methods of the Year winning technologies. And in at least one area, spatial transcriptomics, Scilab Lab has really been leading and driven a large part of the development of these technologies. But together, all these platforms, of course, now produce large quantities of data across all sorts of different data types, which need to be used and utilized and integrated in a medical context to predict and to be combined and integrated. And that is something that Scilab Lab in its current form, and certainly not the academic ecosystem in Sweden, can single-handedly achieve. And that's where the data-driven life science program now comes in as another pillar of Scilab Lab to deal with the increasing amounts of data on the one hand and the rapidly evolving scenery of and environment for data analysis on the other hand. So the aim of the program is really to bring about a convergence of computational and biological sciences. And this is through a massive investment by a private charity, the Wallenberg Foundation, and the Peter Wallenberg here, who's the chair of the Knut and Alice Wallenberg Foundation, announced this in 2020. And the ambition here is a massive long-term investment aimed at placing Sweden among the world leaders in the field and contributing to a better quality of life for people, animals, and nature. So taking a very broad picture far beyond precision medicine, of course. And this was really a visionary investment in my view, because it was certainly thought up and announced long before AI became a buzzword, years before AI became a buzzword, with OpenAI being released. So a little bit about the program. It's funded to the tune of 3.1 billion kroner. That's more than 300 million US dollars, a significant amount for a small country like Sweden. It involves 11 partners, among them all major universities and the Natural History Museum, and it's designed over 12 years to be active in 4 strategic research areas shown here on the right, with basic biology being covered through a cell and molecular biology area, then translation through precision medicine and diagnostics, but also the look to the environment through an area on evolution and biodiversity. Then, of course, inspired by the pandemic that happened at the time the program was initiated, an area to cover epidemiology and biology of infection. So the program is designed to address challenges, among them, above all, the explosive growth of data in life sciences, both production and analysis challenges, but then also the realization that developments in AI are incredibly dynamic, and we need to come up with a program that will still be relevant 10 years down the line, when we do not know quite what the future will hold, except that it's thought that everybody will be able to write code in future, and that AI will have agency and drive robots and move through general to super intelligence, where the AI will be more intelligent than humans. And so how do we deal with that? So in order to achieve that, the program has 6 focal areas. where activities unfold, and each area operates across all the four strategic research areas shown on the left. And so I want to take you through the 6 elements of the program briefly. The first is to attract scientific excellence from a national and international pool in data-driven life science to Sweden. So this involves a cohort of fellows, but also PhD students and postdocs. So over the 10 years of the program, 12 years of the program, the aim is to train and attract nearly 800 or more than 800 scientists to be part of this program. In the first step, this involves attracting 39 international group leaders from an international pool, let's say. Not all of them are international, but many of them are international because we were a bit ahead of the curve. We could recruit some really brilliant young group leaders, and they are active in each of the 4 research areas and based at different universities across Sweden, but all are forming now a community. They are forming, they are very interactive. The aim is, of course, to form a community of experts in this field that connect all the different sites. And let me see, here are images of all the fellows we've recruited so far. And I can not do much, go into much detail, but just give you a few examples here. For instance, Jakob Vogel at Lund University is combining imaging and proteomic data to get at Alzheimer's disease and Parkinson's, or here Wai Wang at KTH in Stockholm is not just pursuing the ambitious aim of getting AI to model a human cell, but he's also keen to exploit AI to design experiments and control microscopes and control the data collection process, which is something, of course, we can think about in the future of life sciences. Then training the next generation goes beyond recruiting fellows. We have started a research school to train the next generation of PhD students in the area. Again, projects have been awarded to all these different universities. The process is that we try to select the best projects. Individual PIs then recruit strong students, and these all form, again, a community of students which are part of a joint research school and take part in shared education and training activities. So we create a training network here, and this will train more than 300 postdocs and students, both in academic projects and in industry collaborations in both cases. So we will create a training hub, or have started to create a training hub, and that will also create a community of trainers across our different universities and of resources for training in data-driven life science and data analysis. And then, of course, we are aware that creating both expertise and services in data science and bioinformatics must be an important part of such a program, because this is, again, something that no individual university can handle, and which would also defeat the point of trying to collaborate and integrate data from different types of experiments. And these are just screenshots of some of the data hosting and sharing platforms that Synapse Lab has. There's, for instance, a portal dealing with pathogens. There's a more broader data serving portal, and data platform that allows scientists to share in one place their data in a way that makes the data findable and accessible. This is supported by significant staff here, and specifically for the DDLS program, for each research area, a decentralized approach has been taken and assigned responsibility to individual universities to support each aspect, each research area of the program with a specific data services and bioinformatics services. And each site is awarded 5 staff to establish the needs of the community and support a specific community. And then, of course, there's close collaboration here with other Scilab lab data platforms in achieving that. And here are just some pictures of the staff that over the very last few months have been recruited into these sites, these data science nodes at individual sites. Here's a view of the National Swedish Pathogen Portal. This has emerged from the COVID-19 portal that was also created in collaboration with EMBL-EBI, and has now evolved into a broader pathogen portal, a data sharing site for pathogen and infection data, being formed into a national portal in 2020, and we continue to collaborate with at the European and international level, of course, because this is more than a national initiative. So ultimately, the aim is here to close the gap between the individual researchers in different universities working in the different research areas and national computational infrastructure, going all the way from bioinformatics support to supercomputing infrastructure. Okay, and then of course we have a need to bridge the gap between life science and data science communities. There has been a previous strong investment by the Wallenberg Foundation in data science and in the computational and also industrial side through the WASP program, the Wallenberg program for autonomous systems and software research. And this has been an even bigger funding program, as you can see on the left. And the aim now, of course, is to interact, for the two programs to interact and to bring that significant expertise to bear on the life sciences by funding joint projects that are supported from both programs jointly. And together, this has led to now, here these 12 or so projects in the first call and a similar number in a second call. So these are all projects where there's a PI from each of the two programs contributing. And it's very important that we find projects here where both the computer scientist and the life scientist actually pushes the boundary and benefits from the collaboration. And then, of course, we are aware of the need to also create partnerships and monitor and push the impact on society. There's an element here that I could have mentioned, and I would have had it not been covered by my anyway, you will see, which I don't see, that there's also a WASP-HS program. This is a humanities and social science aspect of the WASP program, which considers ethical and legal implications of AI, very important, of course. And this also is undertaking joint projects to get at the impact of AI in the life sciences and precision medicine and examine ethical and legal aspects of that. But of course, there are other societal impacts that we need to consider. And for instance, in the infection area that I'm familiar with, we are collaborating with the Public Health Agency, and you've seen the industry partnerships that are part of the PhD and postdoc programs. And then, of course, we are aware that there is significant impact then of these data on for translation, and there's just a little list here of important areas where in precision medicine where AI was recently at the heart of Nature Science publications, all the way to AI chatbots discussing with patients. So these are, of course, all within the scope of our program. So this is where I would like to leave it, and I'm happy to answer questions as far as I can, but please remember I'm really just here on behalf of Ollie presenting his slides, so I might not be able to answer all your questions. Thank you very much.
Yeah. Okay, thank you very much for this really highly informative presentation. And indeed, since you are online, so it's better to ask the questions right now. So is there any question from the audience? Okay, I would like to ask the question. You showed that your system or your program really covers really different areas of research. And when it comes to data, then in each of these areas there are several technical, but even more importantly, different security or data protection requirements. How you deal with that? So is there something that you specifically already have in place that like data sharing is dependent from the domain you obtain the data, or how that works in your case?
Yeah, I'm not an expert in this area, but I'm aware, of course, that this is problematic, and Sweden is not a really good example for easy data sharing of clinical data, because the many regions of Sweden are posing challenges there. And the DDLS program is, of course, a small element in a national conversation to facilitate data sharing. But it is only one player in that national conversation. And everybody's aware that this is a challenge that needs to be solved.
Okay. Any more questions? Okay, if not, then yeah, please. Thank you again for your presentation.
Thank you very much.
And now we can move to the next presentation, which is given by Andre Uitterlinden, who is a professor at the Erasmus University Medical Center. And has been engaged in many genetic studies, GWAS studies, an expert on that. But most importantly, now Andre is leading one of, I would say, one of the, probably that would become a historic actions in Europe, the Genome of Europe project that is really part of this 1 Million Genome Initiative. And I think that can really bring a lot of changes in the area of data availability in genomics. So, the floor is yours.
Well, thank you very much for this kind introduction, Janis. And of course, many thanks to the organizers for inviting me to this beautiful place. And also for yesterday evening's speakers dinner, which was really fantastic. My talk will be mainly about data generation, actually, rather than the previous talk about how to handle the data. As you can see here, this is always a journey of searching, deciphering, and implementing. So we'll speak a little bit about the Genome of Europe program, and at the end I will spend a few words on what we are locally doing at the Erasmus Medical Center on trying to implement all this data in, let's say, a clinical health setting. My disclosures, I sometimes get invited and companies pay for that, but I receive only one salary from Erasmus and I do not hold any stock. Well, this is of course the starting point of many of our journeys, scientific journeys, is that we differ a lot from each other. Look at your neighbor. And that's of course, as you can see from these fine specimens of the human species, not only in the way we look, but also the behavior we have. But of course, for today, most important also the type of diseases that we develop. And what we've learned, summarizing a lot of research over the past 30, 40 years or so, is that variations in the genome, DNA variations, are in fact causally involved in these differences, meaning they do not explain completely those differences, but they have a big contribution to that. And those phenotypes range from developmental phenotypes, so from birth onwards, the way we look, appearance, and as you know, that has had major societal implications in the forensic scientists. Our behavior, also the psychology groups are taking up interest in genetics now to, let's say, create better science from in their discipline. Aging, but maybe more important, the risk for all diseases we know has some kind of background in terms of genetics. So genetic variation is determining to some extent how people develop these diseases, at what age and how severe they will be. And all of these developments have led to this concept of precision and personalized medicine, sometimes called, which is the topic of this great symposium. So sometimes I get the question, it's all about genes, what about the environment? Of course, yes, it's always a gene-environment interaction, so it's not nature or nurture, it's always both, it's an interaction between them. And I like to illustrate the problems we have with those environmental factors with this slide. You see here me enjoying a typical Dutch lunch, very unlike what we just had here, which is usually a sandwich with cheese and some milk, and that's all fine. And that leads to us, as you can see at the bottom, Dutch people having the highest dietary calcium intake in the world. It's more than a gram per day. And that's all fine, but if you travel a little bit south to a country called Belgium, it has very similar genetics, in fact, but as you can see here, the dietary patterns differ quite dramatically from what we have in the Netherlands. And as you can see, I've been the victim of that myself. And that's all fine, but the result of that is that they have half of the dietary calcium intake of what we have in the Netherlands. So same genes, very different environmental factors. So yes, this must have an influence. But I show this slide to illustrate the problems we have in getting hold on those environmental factors, because as you can see here, I forgot how many beers I had at that day, and maybe I mixed it with some shots of vodka or wine. And what food did I have after that? I have no idea. But this is the way how we collect those data. We ask people by interview. So collecting dietary data in this particular example illustrates very well how difficult it is to get proper, accurate data, as opposed to getting genetic data. We can sequence a genome in less than 24 hours now at a very high accuracy. So that's why there's a lot of attention on genetics and much less so on environmental factors, not meaning that they are less important. Again, we are helped a little bit by genomics because now we have microbiome studies helping us, and maybe microbiome profiling from our intestinal microbiome can help in providing some of these answers about what kind of food did we had, where did it come from, what kind of viruses were included in that food, etc. So again, genomics might help us here to get a hold on the environmental factors here. This slide summarizes the type of technology that we've been using in the past decades to find those genetic factors that are underlying those phenotypes and risk factors. And as you can see here, it's a function of the frequency of that genetic variation and the effect size of such individual variants. So you see here on the top left the typical Mendelian, rare diseases, very strong genetic effect, but ultra rare sometimes. And now we have a good technology by sequencing, exome or whole genome sequencing, to find the causal variant in a matter of days now, if you have the proper family material all available and the good phenotyping, of course. On the right side of the slide, you see that the common variants, until not so long ago, were a real problem to identify their contribution to, in particular, the common diseases, which are, of course, the big clinical problems in hospitals. Those are the diseases that clinicians treat on a daily basis. But luckily also technology came to our help by providing the genome-wide association studies in combination with genotyping with arrays, which led to large amounts of genetic data. And in collaborative studies of many cohorts and biobanks in consortia, we were able to identify thousands of these potential DNA variations that contribute to these risk factors. So on an individual basis, these effect sizes are really small, but if you combine them now in polygenic risk scores, and I'll come back to that at the end of my talk, they provide a very strong genetic risk factor, which is equally strong as what you would see in a Mendelian disease. So don't underestimate the strength of their predictive power. Again, this has led to, let's say, a whole library full of genetic variations for hundreds of diseases and risk factors. And now they are bundled in these polygenic risk scores, PRSs, which are now investigated into their clinical utility. Can we use them in a clinical setting, or maybe even better in a prevention setting for making use of them? So this leads, stepping back to, let's say, the scientific approach to genetic architectures we have of phenotypes and diseases. And it usually looks like this. These are typical Gaussian distributions, as you would see here, with effects being very small or very high, frequency being very low or very high, And you see that, let's say, the most common variations contribute to the biggest amount of variation explained on a population level. And those are encapsulated now by the polygenic risk scores. Whereas you go to the ends of the distribution, those are the typical Mendelian diseases, which have very strong effects, but are very rare on a population basis, and do not contribute substantially to the population variance you would see at large. And these pictures hold true not only for the risk factors like cholesterol level. Everybody has a cholesterol level, and everybody has a set of genetic variations contributing to that variation. And the same also for glucose or bone density for that matter. But it also holds true for more concrete phenotypes like diseases, like type 2 diabetes, osteoporosis, and many of the big cancers. And below you see all the technologies that have helped us to identify those genetic factors. So all in all, this has led us to change our view from the genome rather than focusing on one individual variant that is causative for your phenotype or disease to a much more holistic view. So every person, of course, has a whole genome that contributes to the phenotype in that person. So it's not only a matter of a single Mendelian mutation expressing itself, but it's the whole genome context contributing to, for example, the penetrance of that mutation. So these polygenic risk scores not only find their way now into, let's say, preventive screenings of the population, but also in clinical settings to better understand penetrance of Mendelian mutations. Coming back to the sequencing efforts, what has led to all these knowledge about variations? Well, that has coming from massive sequencing efforts. But massive, what is massive, you can see here. We have about 8 billion people on the planet currently. We have sequenced about 5 million people. So that's an ultra small percentage of the people around here. So we've uncovered only a very, very small proportion of genetic variation. But already that tells us that about 500 million positions in the genome can vary between individuals, 500 million. So that's more than 10%, and that's really tremendous. We usually underestimate that difference. People think that we are 99% identical. We're not. We are very different, also on the DNA level. Another point to take home is that it's not only these single nucleotide polymorphisms, or SNPs, that are in there in the genome. It's also bigger variations like insertion deletions or copy number variations. And these very difficult ones, the repetitive sequences, usually encapsulated in the term variable number of tandem repeats, and they also come in different shapes. So variation is also very varied. It's not only the single nucleotide polymorphisms that contribute, but they have become the workhorse of current day genetics, and they are also massively present throughout the whole genome. If we look at how was this genetic data generated, this is a summary slide I made based on public data, but also interacted with companies who sell stuff to you maybe, to generate those data to see how many samples, human samples, have been actually subjected to genetic analysis. And that's shown here. Well, you can see here again the sequencing, so about 5-6 million people have been whole genome sequenced, and the data is available and approachable. But you can see there's a very big difference with array data. So there are over 100 million people on the planet at the moment who have array data. About half of this is in commercial hands at companies until now. Sometimes they collaborate with us in consortia, but sometimes not. And the other half is in academia. And luckily it's also present in these consortia of collaborating institutes, so we can use that to identify those factors. But the major workhorse for genetic data is array data, it's not sequence data. And I think this picture will look like this for the coming 10 years, actually. So this brings us to the European efforts in this basis. So as a start of what Janis introduced, the Genome of Europe, we first started to ask around, what kind of data is there in terms of array data and sequence data. And what you can see here is that it's a very uneven distribution, with the Scandinavian countries and especially the UK being in rather good shape. So a substantial proportion of their population has some kind of genetic data. And of course there are some shining examples like Decode on Iceland and the Estonian Biobank here. But you see the rest of Europe is really lagging behind. There's really a paucity of genetic data there. So the conclusion after making this inventory, which is still being made together with Nicole and Taras from Ukraine, is that we need to sequence many, many more samples in Europe. So this was a good start, of course, for what is called the 1 Million Genomes Initiative. So this is a, let's say, written statement by countries in 2018 when they declared that they would make, in the end, 1 million genomes accessible for research and applications in the clinic. Many countries have joined this initiative, and as you can see here, there are some working groups that currently discuss many different aspects of making those 1 million genomes accessible. And those groups include, let's say, quality and standards for what kind of data should we accept to be present in the database. But also there are some use cases, we call them. So how can we use this data, for example, to resolve cancer cases or to look at rare diseases, but also looking at common complex diseases. And from this effort, the Genome of Europe started, and that's what I will explain to you in a little bit more detail now. A big decision in the beginning was made, okay, 1 million, that's a lot, because the rate of generation of data in the clinic, for example, in the Department of Clinical Genetics or Oncology, was rather slow. So we said a much faster approach could be to create reference genomes from cohort studies and biobanks. So this idea was launched, and then it was decided, let's split that 1 million in two. We have 500,000 reference genomes, and the other 500,000 will come from clinical activities like clinical genetics. So I chaired that working group together with Andres Metspelu, my dear friend from Estonia, the Estonian Biobank, and it was finally approved by all the member states in September 21, and that's when we could kick off, if the slide progresses. I hope. Can you forward the slide? Yeah, there we go. So, of course, a big reason we had to give behind is why are we doing this? Why are we creating reference genomes? Well, a major thing was that Europe wanted to become a little bit more independent in terms of depending on, for example, the US or UK or other countries and create their own genetic data sets. So that was a major driving force. The other one was the European data present in those data sets was really not representative of European genetic diversity. Many countries were not involved in that. And last but not least, it was also initiated as a push to stimulate countries to set up their national genome programs. And that really has been also a successful story. So those were main reasons to start this reference genome collection, so that each country would have proper reference genome data to tell what is normal and what is abnormal in there, and what are frequencies of variations in those countries. And also study, let's say, population subgroups in those countries. Do they differ in composition of their genome? And that also rose to the idea that why not do this on a European scale, because then we can profit from each other. For example, we have German people living in the Netherlands and Dutch people living in Germany. We can sequence our own genomes and make our own national genome programs, but why not collaborate? That we use the German reference genome for the German people living in the Netherlands, and that they use the Dutch reference genome data for the Dutch people living in Germany. And that holds, of course, true for many population subgroups across Europe. So all this together led to the idea, okay, let's create those reference genomes, but why do we need it? What is the purpose of having reference genomes? Well, that's illustrated here. A very simple can be just lookup. So lookup for variations in allele frequency of particular variants across European populations. Let's say lactose intolerance genetic variations or particular breast cancer predisposition variations. How frequently are they across those populations? Does it have implications for clinical care in that country? Another is, of course, to much better understand genetic diversity across Europe by doing principal component analysis. Importantly, also, a big purpose will be the recalibration of these polygenic risk scores to the local population, which is really of importance, coming from, let's say, realizing from the source where they were coming from. And the last one is a more technical one, to provide imputation reference panels for the experts. I can discuss that with you afterward, but it goes a little bit too far to do that right now. So some basic characteristics, what we're looking for in the genome of Europe are normal control people, let's say citizens of your country, of your populations. We really are focusing on getting an overview of genetic variation across Europe, and that means across borders. Also looking at populations which are not represented by a country or a state in the European Union, for example. Take the Sami people in the northern part of Scandinavia, or the Roma people in the mid-European countries. Big subpopulations not represented by a country. We ask for minimal phenotypes, so we're not asking for a clinical bucket full of phenotypes which can immediately be used in the clinic. We start simple and easy and slow with just going for age, sex, and country of origin, because that alone will already be an effort to get such a data set together, giving all the LG aspects, also just touch upon how do we exchange data across borders. We can all imagine the problems associated with that. We will be technology agnostic, so good news for us, we will, let's say, accompany different sequencing technologies, short read and long read in the database. This will be a bioinformatic challenge, but it will also be closer to what will happen downstream in reality. People will be working with different sequencing technologies, so we better be prepared in how to handle those data and try to combine them. We do aim for clinical grade genome, so it's 30x coverage for the experts that we want to include in the database. And as I said, we will do this across countries, so we want to stimulate national genome programs to arise, meaning also that there will be many sequencing centers that contribute to it. And that's very unlike, for example, the UK model, where there was one sequencing center generating all the data. So this was, let's say, the start of it, and that led to this, let's say, distribution of samples. So you can see here at the bottom, it's a number about 500,000 here, and here you see the different countries that will contribute to that. And we propose that that contribution should be proportional. So Germany, big country, is expecting to be delivering more genomes to the Genome of Europe than Malta, a very small country. And how that pans out in numbers is shown here on the right. And here on the left also you see where do we think those samples can be coming from. As I said, we are looking for normal control people. So the best would be population-based epidemiological cohort studies, because they are the most representative of local populations. Biobanks, of course, will also fulfill that criterion. A little bit less optimal is, let's say, taking samples from the clinic, which is not involving patients themselves, for example, parents or relatives from such patients. But all of those will be included in the Genome of Europe database in the end, and some of them will be flagged by that. That eventually panned out into this big table. It's probably not possible for you to read it, but it's not so important. The thing here is that we asked all of those countries to provide us an overview of the composition of populations in their country. So, for example, in the Netherlands, we went to the Central Bureau of Statistics and said, how does our population look like? We have Dutch people, of course, but we also have Turkish people, we have Moroccan people, we have people from our former colonies in there, from Suriname, for example. And all this led to a percentage-wise hierarchical list of composition of the countries. And we did it for all the countries in the Genome of Europe. And that led to this big table. So you see here in the columns all the contributing countries. In the rows, you see the population subgroups based on country of origin that we identified. The white ones are from the participating countries, but the blue ones are from, for example, here, Albania, Algeria, China, Kazakhstan, Morocco, etc. Countries not present in the European Union, but of course, which have many inhabitants in the European countries. So, for example, Turkish people are living in Denmark, in the Netherlands, Germany, etc. So in our efforts to be representative of the European genetic diversity, we will create, in this case, a Turkish reference genome for that matter, by combining data from all those countries. We are, of course, reaching out to Turkey, which wants to become a member of the European Union, to say, can we collaborate? Because they also have a national genome program. And they said, yes, they will collaborate with us, so that's great. And we're trying to do that with all those blue-listed countries here. So this table is a very important, let's say, tool that we use in making sure that what we will generate in the database will be representative for the European genetic diversity. So this is where we are, because there was a lot of talk, of course, about how to do this. Then a call for proposals was opened, which in fact was only one party was eligible, namely us, our working group. And we were awarded in April this year a contribution of 20 million euros by the European Commission. But in the Digital Europe program, in which this project takes place, they ask you to match this amount of money. But it went very well, because we have over more than 20 million matching funding. So the total budget now is 45 million euros, which will be spent on mainly sequencing genomes from all those different European countries and population subgroups. As I said, it is proportional and it is representative. And what we also notice is that we now have 31 countries participating, so that involves 26 EU member states, but also 5 non-EU member states. You see them listed here. Serbia, Norway, Turkey, Ukraine and Moldavia. They also expressed big interest to join this effort. So we're doing our best to accommodate that, of course. That comes with some challenges, because not all countries are part of the European Union, but also they are not part of this Digital Europe program. So that will require some more effort in order to make them really part of that. But we are already involving them in our discussions, for example, about sequencing standards and how to integrate such data. Well, here you see in the balls the type of work packages that we have, looking at many different aspects, of course, and I will not go into the detail today, but we have several working groups there addressing them. Well, this is, of course, in the end what we would like to create. We want to recreate a map of Europe, but now based on, let's say, genetic diversity. And in fact, an effort to that, not based on genetics, was made by a guy you might know, Freddy Heineken from the beer, Dutch guy. And in the 1990s, he reached out to some historians, and he wanted to create a map of Europe based on ethnic background, on language, on cultural diversity, etc. And that led to this fantastic map of Europe, which of course has much more grain detail than the map that we know based on country borders. So this in total had 75 small member states, according to Freddy Heineken. However, you also know that reality is of course much more challenging, and that's shown in this slide. This is a map of Europe based on prejudice that we have among each other. So, for example, you see here that Holland is in here for Sodom and Gomorrah, so anything can goes. You have here the smelly people in France, Mexico, the commies, the buffer zone, etc. This is a map, by the way, made by a Bulgarian artist, Janko Svetkov, and I'll invite you to go to his website. They're really hilarious maps. And when I travel to the States, I usually call this Europe according to Donald Trump, and that's always well received and recognized. And this is, of course, what is the actual case, and how to get all these people together. And on top of that, politics is not helping us. For example, with Brexit, it was suddenly impossible for us to just collaborate with our dear friends in the UK and get access to their fantastic data, because they were way ahead, of course, of what we are now doing in Europe. So politics is really not helping us. And then we have internal problems, like in Catalonia, in Spain. They want to become independent. So should it be a separate country, yes or no? Maybe Scotland will join them. And then we have Turkey, I already mentioned it, wanting to join the European Union, but there are political hindrances for that. So yes, the politics is not helping us here, but we are doing our best to reaching out to those friends. So you see me here with my friend Matt Brown, who's now the Scientific Director of Genomics England, and Rory Collins from the UK Biobank. Of course, we have close collaborations already, but we have to find ways to overcome all these political hindrances. The same holds true for the United States. I was fortunate enough to meet President Obama at that time to discuss the All of Us program and how to interact with the Genome of Europe program. I showed him the map, and it was quite a funny conversation we had about that. So yes, we are reaching out for other countries and setting up collaborations across the globe to have people also allowing access to that Genome of Europe database someday, I hope, under some circumstances. So where are we now? Well, most important, we will have a kickoff in Rotterdam soon, end of October, with all the participants in there. We are busy making the consortium agreement. The grant agreement has been finished. Official starting date is October 1. And we are already having several meetings, for example, about sequencing technology and sequencing standards, how to do that for quite a while. We are in close contact with the technology providers to explain to them what we're doing and to discuss with them how we can make use of their technology. And we hope, of course, also some discounts for the consumables. It also has led to, let's say, a revival of interest from non-participating countries like Poland and Switzerland in the European Union, and some new countries that express their interest. I mentioned Ukraine and Moldavia already. And of course, we're always in lack of money because we have many more countries than we originally were budgeting for. Some accompanying developments are really helping us here also. I won't go through all the list here, but especially I'd like to mention the Draghi report here at the end, which was released in September, and maybe some of you might have read it. It mentions genomics at 3 paragraphs there of the 400-page report. And they really see this as a technology which should be invested in across Europe to help innovation and to help the competitive power of Europe. And we were also very lucky to see that that particular topic of investing in genomics and increasing sequencing capacity ended up in the so-called mission letters that Ursula von der Leyen, as the chair of the Commission, sent to her commissioners. So these are really tasks for them to be picked up by them. So in fact, we are really, let's say, in good shape with the Genome of Europe because we are delivering already to what the Draghi report was asking for. So we're very happy with that. Of course, we also have some ideas about the future. Where will it go next? Well, one thing is we created another working group on pharmacogenomics to also, in a very similar way, get European people together across borders to help compare notes, set up maybe guidelines across the European borders to be able to exchange data, to harmonize the protocols in there, and maybe also compare the clinical guidelines and maybe try to adjust them in such a way that we can easily exchange pharmacogenomics data there. And that's going fine. It's being led by Jesse Sven from Holland and Lili Milani from Estonia. Of course, here there's a whole wish list of other activities we have in mind. I won't go through all the details here, but especially this one, we really want to use DNA information in a very broad sense into healthcare, but in particular in prevention. And that will be the final part of my talk. How can such a thing look like? Well, we're going back to GWAS. What we've seen over the past decades or so is that there really has been a growth in the size of the consortia. So as you can see here, this is a number of diseases and phenotypes, and here you see the years. And here you see, let's say, the number of samples that was involved in the first GWAS that created 210 hits that explained 40% of the variance. And for this particular phenotype height, that increased to 250,000, 700 hits, 30%, and now over 5 million people was published last year in Nature. 9,000 SNPs, more than 50% of the variance explained. So we have here a PRS which is really explaining and of use to use in a preventive setting. But you would say, yeah, what about height? It's not a clinically relevant phenotype, although that's not completely true. But you will see the same holds true for things like myocardial infarction, age-related menopause, age-related macular degeneration. So this development of increasing sample size of GWAS leading to much better PRSs, which become clinically useful because they explain so much of the variance. So here you see what the polygenic risk score is and how we can use it, for example, in prevention screening programs. I don't know how the situation is here, but in Holland we have a mammography screening program for women for breast cancer. So if you're age 50 or older, you will get a call to be part of this screening program, and every 2 years then you are subjected to mammography. How can we change this in order to make it, let's say, more bearable for the participants? Mammography is not a nice thing, I understand. And how to make it more cost effective? Well, the big idea about that is first do a genetic test based on the PRSs, and then invite women based on the risk category for that mammography screening. And that will translate into, if you're in the highest risk category, like for example here, that's a small percentage, 1%, or 5% of the population, but this is a substantial number, of course, on population scale. Those women will be called immediately into the mammography screening program, and that can happen at age 30, for example, so at a much younger age than age 50. Whereas the lower risk category here, they might not have any added value or added profit from doing mammography. It will not increase their risk to such an extent that they will enter clinical screening, for example. So it might not be necessary at all to do mammography for that group of people. And in the middle group, and that's of course depending on how do you define those cutoffs, those women will be subjected to the normal screening program. So this idea is now being tested in trials actually across the world. There are about 4 trials now running, and we expect the first results next year and the year following that, to see how does this improve the screening program and how can we then translate this into the individual countries. I gave you the example of breast cancer, but you see here on top that same approach is now being contemplated for many different diseases. And those are the big diseases of our society, the real killers, so to speak. And they include cardiovascular disease, type 2 diabetes, asthma, etc. So the idea is that we first do a genetic test and then identify individuals in high risk, middle and medium risk and low risk. And the nice thing is that, according to my view, but of course I'm very biased, is that with this one genetic test, you can do all of this. You can extract all of those PRSs out of that. So why not give everybody a genetic test at age 30, or maybe even better, at birth? Do it at the heel prick. So there are big debates about it. We can have endless symposium about this here, about the pros and cons of that. But this is being contemplated now, how to implement this. Maybe take a few of those diseases and examples first, and then see how it works, and then start to expand it. So that's PRSs. A big driving force in there has, of course, been the cost. Because the cost of an array, thanks to companies like Illumina and Affymetrix, has really dropped. It used to be close to €1,000 per sample. Now it's less than €30 per sample. And this is 10 times lower than whole genome sequencing. It also creates 10 times or maybe even 100 times less data. And the big nice thing, I think, of arrays is that it will only provide you with useful data, as opposed to whole genome sequence data. You will be drowning in unuseful data which we cannot interpret, but which might need follow-up. So these are a lot of false positives in there. So that's why many people are thinking about array genotyping as a genome-first approach, for example, in such population screening programs. But we're also doing that in the hospital, in Erasmus Medical Center. But before we could do that, there were some technical challenges we had to overcome. For example, the question, can you actually detect Mendelian mutations with an array? Well, the answer is a full-blown, yes, we can, quoting Obama. Because we showed that already for a number of diseases in which we collaborated with the clinical departments, for example, of immunology, of clinical genetics, and of cardiology, to show, for example, you can detect all the hypercholesterolemia mutations present in the Netherlands on a single array test for less than €30. We had a program screening for FH carriers, but that was stopped because of the cost of the sequencing. And now that is being revived because of the cost of genotyping is much, much lower. But now it will be done with arrays. As you can see here, we have a number of other manuscripts now in the pipeline, and in every case we can screen those Mendelian mutations. So yes, you can do that. So that leads to this conclusion. This will be a very cost-efficient genetic test to be entered into the clinic, but also in these population screening programs. And that led to these, let's say, potential applications. I'll disclose some of them to you already. For example, the FH screening program. We're actively discussing this now with a group in Amsterdam, how to set that up in the Netherlands. We're also talking to other countries who have similar programs stopped because of the cost and are now rethinking that. Of course, pharmacogenomics, also a no-brainer. Everybody wants it, but cost is prohibitive. But now for less than €30, we can provide a very accurate pharmacogenetic report, including 2D6 for the expert. Very well. Mendelian mutations, blood group typing. We're discussing that with the blood bank in Amsterdam again. Maybe those blood group typing can be done much more efficiently and extensively with arrays, and then maybe also include HLA typing for that matter. And then I discussed it with you already, the screening programs. Now for breast cancer, but of course many other major cancers which also have screening programs. And this is my final slide because that led to an activity we have at the Erasmus Medical Center. It's called the GOAL project, genotyping on all patients. For clarity, we are not doing that yet, but we are setting up and have set up pilot projects with a number of clinical departments to discuss with them how will that look like in your case. If you get a patient, before you see that patient, a genetic test might be informative for some of the decisions that you have to make. So what kind of genetic information would you need? Those could be, let's say, some Mendelian mutations you want to exclude from the diagnosis, so it's not a very rare disease we're looking at here. It might be ancestry information you would like to have, or maybe pharmacogenetic information for the treatment, etc. And maybe PRS is, of course, also there. And that leads to this simple picture that when the doctor sees that patient, a very simple screen, green, orange, and red, will tell him or her, you have to act. If it's red, the screen will block, and unless you deal with the genetic information, you cannot proceed. If it's green, nothing to worry about, but you can click on that to get more information about it. And that will help the doctor in setting the diagnosis, designing the treatment for that, personalized, more precise, and also maybe provide some lifestyle advice. We've discovered, for example, somebody who came in for breast cancer that she was in fact an FH carrier without knowing it. And that might also be beneficial for that patient. So that patient will leave, of course, cured and all, the hospital, but now with genetic information which is useful also for the rest of her or his life. So these are ideas we have. The pilot projects are going quite well, I must say, especially with the eye clinic. They are using it in a clinical setting already. Clinical genetics is using it now for breast cancer screening in the hospital already. And the FH I told you about, that's done with the Amsterdam clinic. So slowly this is growing, and now the final step we have to take is to convince our board of directors that this is really useful to invest in, and to convince all the department heads in Erasmus, which is not a tiny thing, I can say, to start doing this. But I'm very hopeful about that. So with that, I would like to thank you for your attention, not without highlighting these people. I'm always standing on shoulders. They are doing the hard work in the lab, and I'm just here telling you about it. So thank you for your attention.
Okay, thank you. Okay, I guess we can leave the questions after the last presentation, and we all come here on stage. But okay, the last presentation is one that I have to present. Okay. Okay, and I'm really happy to provide you with some information on Latvian case, and so this will be in part also the data that we are collecting in frame of this really huge initiative, 1 Million Genome Project. But I will also try to give you some examples where we already start to combine the genetic data and also medical data to really look what we can do to help also influence some healthcare processes and applications. So to come to this stage that we can actually call ourselves a genomic database, or we have some huge data, it needs a lot of preparement. And here I can show you some milestones, what we have done in Latvia, that started with some preparation of legislation, establishment of infrastructure, most importantly, National Biobank, and also not forgetting to join some international activities where we can share the data, share experience to really be on the right track. And finally, that has led us to really the larger initiatives where we can now start to really talk about the Latvian genome reference as something that is that will very soon be established. So let me first start with a few infrastructure elements that we have here in Latvia. In 2005, we established Latvian Genome Center that has two important functions. We are doing the biobanking and also providing the sequencing technologies. To talk about our biobank, this indeed is one of the largest biobanks in this area of Europe. We are really working as a service provider for many projects here in Latvia, and that has led us to the stage that we are not only the population biobank, we are not collecting only the random samples from the population, but we also have a good representation of different disease cohorts that can be studied immediately. And so we also have worked hard to, without the much experience and also probably not yet with the optimal legislation, to perform the data retrieval from different national health information systems. So some pictures from our infrastructure, and I'm really happy that during this road we can we have a possibility to discuss these things with the technology providers and different private partners. And here I can only mention that indeed we now have a possibility to do the different and also the high throughput sequencing using different technologies, including MGI and very recently Oxford Nanopore. And especially I would like to thank MikroTik that has really helped to establish because of course the data generation is only one part, then the data analysis, data transfer is something that we cannot live without, and that is really done with a big help from MikroTik, who really prepared this genomic data network and also helps to increase the capacities at the high performance computing center in Latvia. So what is Latvian genome reference? Andrei already showed you the reasons why we need the reference, so I can basically skip this argumentation why we need that. But of course we see this also as a part of this 1 million genome initiative that Latvia joined also in very early in 2018. And in our case, we just wanted to be on a safe side. If you looked at this data that Andrei was presenting, our share into this 500,000 European genome reference would be 2.3 thousand individuals. We decided to do a bit more. So basically what we are now really intensively sequencing is establishing a population cohort of 3,500 individuals that covers different, the people come from different Latvian regions, and they can apply themselves to this project. And also, so at this stage we have some, almost all the approved participants are here, but still there is some place for more participants. And also we do the sequencing intensively, and again we have to finish this project till the end of the year, and this is very close, as we already have sequenced 2,500 whole genome sequences in our facility. This project is funded by the Recovery and Resilience Facility, and of course, as already Andrei showed, it kind of precedes this Genome of Europe project, where our sample can be used as the legacy sample. And we can also, I think we can be very ready to provide these samples if there is need for some testing how the data are used or analyzed. As I mentioned, participants are involved from all Latvian regions. We have agreements with the laboratory, with the Gulbi laboratory, who is performing this recruiting of the patients, and there is also online form developed so that to make this process easier, and we are really happy to all the participants that has already joined the project. And of course, the most important thing is to how we can deal with the data infrastructure. Here I'm showing you the data flow, and I'm really happy that we had this opportunity actually to host the central data storage in this house. So basically the Latvian National Library is not only the place where the old books are stored, and also we are having this conference, but it is also one of the approved data storage centers in Latvia. So and this is the place where we will store. So this it is a real, our data is a real value also for the country and for the history as well. So, of course, I'm not going to go into the details of all the data flow, and some of these pipelines are all on and processes are established already. Of course, we need to use the high-performance computing cluster that is located at the Riga Technical University, and the next steps would be to really understand how we can make these data available, and this is again, all this process is covered by participation in different projects. So, as I mentioned already, the sequencing part is covered by this local RRF funding, but we are now starting the Digital Europe part of this Digital Europe project that we will also bring some more data in. Another part of this RRF funding goes for this infrastructure establishment that I'm presenting now, and of course to ensure that these data can be properly assessed by the other researchers in Europe. We are also participating in the European Digital Genome, in the digital GDI, Genome Digital Infrastructure Project. So more details on this system. Initially, of course, for the genome reference, you would not need much. So basically, you can do a lot with the aggregated data, see the frequencies of different mutations and so on. But we established this project in a way that it can grow into the more important storage system for whole country, where the whole genomes can be stored at this location. And of course, for that you would need to have a proper access elements. So basically, this is still the conceptual architecture, but project is started, and we have this decided that all the participants can access the data using this ADAS authentication, and also we are working to establish different work environments that may be not needed for this genome reference project, but that can be used later on, so that actually each participant can access their data individually, and also provide the kind of possibility to do it from the doctor's or researcher's point of view into the regulated framework. So, but of course, this is just an infrastructure, and I would like to really share you some information on the analysis that we already have done with the first genomes. And of course, the one thing that we started to look at, what is the Latvian genome variation? How do we look? And in such studies, you always find new mutations. Andres showed you that how many different sites are mutated in our genomes. Of course, if you just add a couple of 100 people, you will find definitely some variants that are not described, but that, of course, is just because of the natural variation. And what we already showed that for the technical imputation panel, that if we use our data in this process compared to the global data, we can increase the precision, we can increase number of variants called especially for the low frequency and rare variants compared to this global reference panels. So basically meaning that indeed each country has to have this genome reference to be more precise in different steps of the genetic analysis. Of course, this is just another thing is we just started, but we will definitely look for the most frequent pathogenic variants. That is something that we can help also to the rare disease community to understand what is this background in Latvia. Of course, one interesting thing to do is to compare our genomes with others, and this is the picture where you can see where the Latvian sample, which are these these dark squares located on this variation map. And with the gray sample, you can see the samples that are ancient genomes, actually something that has been analyzed from the barriers or from the archaeological sites. And as you can see, that these also include some samples from Latvia, dating even 7,000 years before. And as you can see, that we are we are the population that is most related to these samples, obviously, because they are from the same territory. But interesting that according to our analysis, the Latvia represents a population with the highest degree of this ancient Western European hunter-gatherer genetic ancestry. And we were kind of probably isolated from the other influences and genetic exchanges from these Anatolian farmers or probably steppe people that came to the rest of Europe. But this is something that we are continuing to investigate. And now I'm coming to the examples of using genomic and medical data together for some medical needs. And the first sample I would like to show you is we're going to test whether we can use these polygenic risk scores, in this case type 2 diabetes polygenic risk scores for type 2 diabetes prediction. And as already was mentioned, that indeed it's not, and we just discussed it also yesterday, that it's difficult to use the model, the polygenic risk score calculation model that is calculated on different population. And for that reason, we actually decided to take the all-known polygenic risk scores, more than 100, and compare them how they perform in our population. And if you first thing that we can see that indeed these models improve with time, because they can explain more genetic variants. So comparing like models that are developed our days compared to those that are developed 10 years ago, that's of course because of the data that has been used to develop these models are much larger. And we decided to choose one of the best performing model for our further studies. And actually, this is the model that comes from the Finnish population, from the FinGen, that performed the best in our population. We also actually developed the type 2 diabetes polygenic risk score that is from our own Latvian data and discovered that it has the same performance as the model I was just talking about. Another thing that probably many of you know that Latvia's demographic structure actually underwent a significant change due to Russian occupation and followed by a massive wave of immigration. And then we decided to look whether there is a difference in ability of polygenic risk scores to predict the disease, taking into account that fact. And we analyzed across two major population ancestry clusters. We identified them unbiased. And indeed, one of them corresponds more to the people that reported their nationality, because the self-reported nationality, of course, or ethnicity, as Latvian, and other was more reported by, as you can see, Russian, Ukrainian, Belarusian, Polish, and so on. But of course, the good thing is that this polygenic risk score performed constantly well across both groups, and that means that we indeed can use these polygenic risk scores in our population with some degree of probably we have to be careful and compare these different risk scores for different diseases. And the real application where we decided to test it is to look whether we can differentiate between the different groups of patients. And in this case, we decided to test a cohort of monogenic diabetes or early diabetic, more correctly, that these were early diabetic patients that were involved in our biobank, more than 66 people, for whom there was no specific genetic diagnosis established. And we decided to use whole genome sequencing in this case, so that allowed us both to look for the rare mutations that can explain or assign them to this MODY-type diabetes, and also calculate a polygenic risk score. In this cohort, we identified 26 causal variants corresponding to different types of MODY diabetes, but of course this still left quite a large proportion of patients without a causal variants, and we decided to look whether we can see the difference in the polygenic risk scores for type 1, using the type 1 diabetes and type 2 diabetes polygenic risk scores. And we also used control group in this study, and also type 1 diabetes patients and type 2 diabetes patients, and all of these patients data are coming from the whole genome sequencing. The best results we of course got with the, which has already been described, with type 1 diabetes polygenic risk score, which can, as you can see, that can, first of all, that can clearly differentiate between the controls and type 1 diabetes patients, but we also see that there is a gradual increase in polygenic risk scores if you look at the patients who do not have these polygenic monogenic mutations. And actually, if you take the top 25 percentiles of the polygenic risk scores, you can see they are mostly populated with the patients for whom we were not able to identify the monogenic mutation. And even more, if you look at the insulin usage, which is prescribed in this case, not based on the genetics so more or less empirically, so that we see that indeed this that again proportion of the insulin users at this higher polygenic risk part is really significantly higher than non-insulin users, again meaning that most probably these are type 1 diabetes patients that has not been identified before. So, as one of the consequences of all our activities, we are also developing the polygenic risk score calculator that can help us to really to use this information more broadly. And actually, this is something that we are going to open this next or probably month or end of the year. And the main features here is the ability to submit both the array genotyping and the whole genome genotyping data that has already been adapted to Latvian population. And so we make it in a way that it is, we can customize the design, so that we can create a packs with the chosen set of models that depending on the needs of specific specialists or for the research needs. So this is something that is already working. We are testing these first packs of different polygenic risk scores. Another example that I am more turning not only to the genetic data, but this one is an example where we compare different multi-omics approach and identify the data from the medical records to identify the molecular footprints of COVID-19 patients, especially in case of long COVID-19 disease. So basically, this study is based on the patients that we have involved at the beginning of pandemic. We involved more than 600 patients with really many different samples, and especially for those that were hospitalized, we also have follow-up, at least 2 follow-up stages, where again a large set of different samples, including the blood, urine, fecal sample, and other samples were collected. On the other hand, to really understand which of these patients have long COVID diagnosis or which suffer from the long COVID, we use this different data from the electronic health records according to developed strategy that is published in this paper, and we looked. So basically, we were able to identify the long COVID patients from the electronic health records, and that led us to this design where we can really actually study both. So basically, with this longitudinal sampling, we can answer to the question how hospitalized COVID-19 patients recover, looking through the data longitudinally, and also perform cross-sectional comparison to see what genes or metabolites are involved in long COVID disease. The first study we did with mass spectrometry-based blood metabolomics, and we were one of the first to show that this tryptophan, arginine, and the glutamine metabolism are one of the key differentiators between this acute and recovery phase, and that also we can see that for example, these metabolites are increased in the patients that are recovering more slowly. Another sample that we, in a similar design, in the longitudinal design, where we actually see that dysregulation in amino acid metabolism and also in markers of dyslipidemia are associated with this acute and recovery states. In this case, this metabolomics is done with the NMR. And again, leading to some hypotheses what pathways are involved in this process, and also looking cross-sectionally between the long and recovered patients, using the blood cell transcriptome analysis. We discovered quite many transcripts and pathways that can be involved in this process. Some of them are showed here. For example, the pathways that are involved as a part of immune response. Actually, for the patients that suffer from the cardiovascular traits, there is also explanation from the low-density lipoprotein receptor, the differentiation in this stage, and so on. So I'm not showing, of course, all the data. And finally, this also helped us to be part of the larger consortium. In this case, I'm showing you this just recently accepted data on genome-wide association study of long COVID, which has helped to find also genetic factors, and using these medical data from different records, we could actually be also part of different international consortium. And with this, I would like to end. So I'm really happy to my research group that I'm showing here. All the people that are really young, enthusiastic, and working on different projects in which I believe also the reference will be used. But of course, I'm really also thankful to all the collaborations we have with the hospital, with the doctors, and specific thanks to the patients and participants that involved in all these studies. Thank you. And for those who has not yet applied to be part of the genome reference, this is Latvian nationality only. Sorry, you can scan this and still, as I said, we are finishing the project. So but you can still manage to be part of that. Okay, but now I ask Andrea to come to the stage, and we have some time to answer questions if you. Any questions from the audience? Okay, yeah.
Thank you. So the question was, how much, do you have any data on how much actually each of the countries is spending on genetic testing annually? Would it make sense to just, you know, screen every newborn, since the genotyping with the arrays is so cheap? So that, I mean, you can use that information throughout the lifetime, and pharmacogenomics is the first thing you can use right from the birth, because you know what medications are going to work and which are not.
Difficult question. I do not know the number, speaking for the Netherlands, I do not know the number because it is so fragmented across different clinical disciplines and across medical centers. So I think it will be quite hard to get a number of that. But what I can tell you is that we did this exercise for Erasmus Medical Center. So we calculated, this is like 4 years ago, to get this goal project started, to show to the board of directors how much money is spent on genetic testing. And that was on an annual basis then about 5 million euros. And we concluded that if all of that testing would be replaced by an array, it would be about 500,000. So they could save money. However, you can also realize that's a simple calculation. Of course, in practice, it is much harder. Because what I illustrate also, you have to negotiate with each clinical department, and that's depending on our healthcare system, which gives a bonus for seeing patients and treating them, not so much for curing them and preventing disease. So the whole healthcare system has to be, let's say, scrutinized in order to see how to implement this. So the cost is, of course, one thing. But I think the whole logistics of the healthcare system is a more difficult one now that we encountered, at least in the Netherlands. You also said you have a very nice compact country, and I'm really jealous about it, because here it looks easier to get something like this implemented in here, because you might have only one laboratory or one ministry dealing with this. So, and that's a problem we have with a slightly bigger country being much more heterogeneous. You have to talk endlessly with all these institutes and people. So.
Yeah. Okay, now, I also don't know the numbers, but I think the biggest problem is, especially for countries where the genetic testing is, you can't save money if you're not spending money. So basically, if you don't have this genetic testing developed, so that it's still it has to be in place, but you cannot use as an argument to really implement. On the other hand, it can be a good argument to start from the right side, so you don't have to repeat the mistakes that somebody else has done. So yeah.
Maybe another point on your suggestion to it with the heel prick and newborn screening. Yes, sounds easy, but it comes with a lot of ethical issues, of course, also. For example, would you like to include a breast cancer PRS in that heel prick and then inform persons about that? Is that wise to do, yes or no? And how will this translate into clinical activity somewhere down the line? So in the Netherlands, that's why we have a very conservative system at the moment. I think that's right, to only look for treatable diseases, curable diseases that will be affecting the child between a certain age window. And then the current thinking is that for those population screening programs, maybe we should move to age 30 or so, when all the brains have matured and people are taking responsibility for their life, they're buying houses, having children, etc. That may be the good moment to also include such a genetic information test for them. So that's the current thinking, at least in the Netherlands. Yeah.
I see there are more questions.
Yeah, it's not more a question, it's more a comment about the newborns and testing. If testing is so cheap, it does not make the sense to test it in the newborn age, because the storage of data, safe storage of data, would take quite a lot of resources as well. So if the testing on array is so cheap, so it's maybe worth to test on the moment when those disorders are really important. You would screen only those which are childhood ones in the newborn age, but those who are adulthood, you would screen later.
To finalize this, maybe another comment is also technology. So now we have arrays and sequencing. What will be there in 5 years? I don't have a clear idea about that. So we have to be cautious with saying, okay, we do a one-time whole genome sequence and that's it for 50 years. I don't think that will be the case. We'll have to redo the whole genome after 10 years, I think, because of all this new technology that will be around. So I would also be in favor of a more conservative approach now.
Yeah, okay. Actually, I was thinking of another question I wanted to ask you. Genome reference, of course, solves some problems, but not all of them. And in case of this, for example, the use of the polygenic risk scores, we still cannot really, I mean, this reference is not enough to really calculate the polygenic risk scores that are specific for the population. So and then still there will be an equal kind of distribution from that sense. Can you, I don't know, maybe then it's idea how to solve this problem without just telling that you have sequence, I don't know, 10,000 more of them.
No, well, I think your biobank is a perfect example of that. So you collect not only the DNA and blood samples, but also phenotype data, and you follow these people for a longer time. So you have information about diseases, etc. Like we have in our case the Rotterdam study doing that for already 30 years. The Estonian Biobank doing that, Decode, UK Biobank. Those are fantastic resources in the long run to collect such data, which will make and will allow to calibrate those PRSs locally. So I was looking at your number, 40,000 samples. Great, let's all genotype them and then recalibrate those PRSs. And that's part of the plan we have now after the Genome of Europe, or it should be going in parallel, to, let's say, open up all the biobanks and cohorts we have in Europe, and there are millions of samples laying out there with lots of phenotype data. We should all genotype them and then start recalibrating the PRSs. Yeah.
I think the best strategy would be not to collect samples if you can't genotype them, because it's a price of collecting more or less. So that is the strategy that we are now following, but still we have many samples that has to be
Please continue.
genotyped. Yeah. Okay, if there are no more questions, even though we had 3 speakers, we are over time anyway. So okay, but thank you very much.